Welcome to the PD Exchange, the Peritoneal Dialysis podcast. I'm your host, Dr. Nikhil Shah, a nephrologist at the University of Alberta, Edmonton, Canada. And with me, as always, is Dr. Jeff Perl, a nephrologist at the St. Michael's Hospital in Toronto, Canada. For the first podcast of 2025, we have chosen the holy grail for PD, ELIXIR in peritoneal dialysis solutions. We are discussing the rationale and the design of ELIXIR, a randomized controlled trial to evaluate efficacy and safety of Xylocore, a glucose-sparing solution for peritoneal dialysis. And we welcome Dr. Arduino Arduini and Dr. Richard Corbett. Dr. Arduini, who graduated from the D'Annunzio School of Medicine in Italy and worked in the US and Italy until 1998. Subsequently, he joined the pharma side of medicine as head of metabolic diseases in Sigma Tau Pharma industry in Italy and as the disease area head of Hoffmann La Roche in Switzerland. He is currently engaged in research and development of innovative therapeutic platforms for kidney and cardiac failure. And we have Dr. Richard Corbett. Dr. Corbett is a consultant nephrologist at the Hammersmith Hospital in West London, UK. He also chairs the London Kidney Network Dialysis at Home program and serves as the NIHR lead for renal research in North London. His center is one of the participating centers for this randomized controlled trial. Welcome everyone, folks, and I will hand it over to Jeff.
Thanks, Nikhil, and it's great to have Richard, and it's great to also have Arduino as well to talk about this. And we're kicking things off in 2025. I want to wish all of our listeners a happy and healthy new year as we kick things off to talk about Xylocore. It may be the magic elixir of peritoneal dialysis. And as we all know, for many years, glucose has been the mainstay osmotic agent to allow us to treat patients with kidney failure on peritoneal dialysis. It has its ups, it has its downs, which were recently debated at the ISPD Congress in Dubai. And it seems to be the solution that we've been using for quite many years, and almost synonymous with PD is the use of glucose-based solutions. So it's exciting to see innovation in the area and us challenging the paradigm. And maybe I'll start off with you, Arduino. Tell us, what is Xylocore? What's it made of? How did you come up with the idea? We want to know all about Xylocore, this new solution.
Yeah, let me start from the very beginning. This was happening more than 20 years ago, and I was talking with a good friend of mine, a nephrologist. At the time, I'm talking about end of the 90s, late 90s, 1999 actually, and I was leading the metabolic disease department in Rome. I was heavily involved in developing anti-diabetic drug, and I'm a metabolic fan, as you may know. But anyway, in talking with Mario Bonomini, actually then eventually became an author of several papers that we published together, and is also, let's say, a PI of the ELIXIR clinical trial. He showed me a leaflet of Dianil. At that time, again, I was not aware even of this modality of renal replacement therapy. I knew about hemodialysis, but I was completely unaware about peritoneal dialysis. And when I saw and I went through this leaflet, I was a little bit, let's say, shocked. I said, Mamma mia, this is loaded with glucose, this solution provided through an IP route in a patient that sometimes is even, very often is even diabetic. It actually loses kidney function because of diabetes. So I thought that was insane, talking with him. And he was challenging me. Why don't you think about it? Why don't you come up with some, I don't know, new ideas or new formulation capable to somehow remove most of part of the glucose and replacing it with, let's say, osmotic agent that should be on one end safe, because safety here is an issue, because you are not dealing with milligram of something, you are dealing with several grams in order to establish, let's say, an appropriate osmotic gradient and to have fluid moving between the two compartments. But anyway, but in my opinion, considering that these patients are very often insulin resistant because uremia lead to is one of the cause of insulin resistance plus diabetes, which is on the background, then I said, why don't we try to select osmotic agent capable to exert some favorable metabolic effect, particularly with respect to the sensitivity to insulin, limiting insulin secretion, because this is another thing that was really disturbing, at least to me, because here, beside the direct effect of glucose, we should not forget that glucose is the most potent insulin secretagogue. So these patients are constantly hyperinsulinemic, and hyperinsulinemia causes insulin resistance. Just for this, let's say a simple tachyphylaxis, post-receptor desensitization, and so on and so forth. A few years later, I left with these thoughts in my mind, and I said to Mario, I told Mario, I will think about it. And then at that time, I was working on the carnitine system, and one of the molecules that came first in my mind was carnitine. And the other thing of carnitine or any other molecule is the fact that if you want to select compound capable to exert, let's say, a favorable metabolic effect, you may eventually need to overexpose insulin target organ to this guy. So I was, again, reasoning as an anti-diabetic, person searching for anti-diabetic drug. So I went through a sort of knowledge-based screening because we know that we knew some, low molecular weight compound capable to exert good osmotic effect. Actually, at that time, I was working on a carnitine palmitoyl transferase inhibitor to modulate gluconeogenesis. The other ingredient that I was thinking of was xylitol. I was following xylitol, particularly from the old German studies in the early 70s, because they were using xylitol as a replacement of glucose in total parenteral nutrition. And I was very intrigued because they were using really large amount of xylitol, exposing, let's say, critically ill patient with this significant amount of xylitol. Xylitol is a natural occurring compound like carnitine. It is produced by the liver up to, let's say, 10 to 20 grams per day, and it comes from the glucuronic acid pathway. And then eventually, xylitol will be recycled within the pentose monophosphate shunt. So it's completely consumed. So then I said, all right, now I have, let's say, a better picture because I can use relatively large amount of carnitine, not that much because carnitine cannot be fully metabolized. Xylitol could be really the main osmotic ingredient, will be fully metabolized to carbon dioxide and water like glucose. It is a very poor insulin secretagogue, and that for me was really a must. Plus, one of the intermediate of xylitol which is formed within the pentose monophosphate shunt is xylulose 5-phosphate, which is a powerful inhibitor of gluconeogenesis. I said, this is the recipe.
So let's take a step back. So just for our listeners, Xylocore is a combination of carnitine and
xylitol and glucose. You have a ternary combination where the main osmotic ingredient will be xylitol.
Okay. So there's xylitol, there's carnitine, and there's glucose, correct?
There is glucose.
Okay. Clearly you're passionate about this, but none of us have the same breadth and depth of understanding as you. Why? So just in a simple terms, it cannot be all xylitol because?
It cannot be all xylitol because we don't want to overexpose, particularly the liver, to xylitol, which may form elevated, higher level of glycolaldehyde, an aldehyde which may be form oxalic acid, and oxalate may precipitate. Oxalum is an example.
Okay. So overexposure to xylitol can increase oxalate. Okay. Now, why can't it all be carnitine?
Then we were thinking to use all carnitine, and I went up to 0.25% of carnitine in the previous study. Then with carnitine, you really overexpose and probably too much, and maybe it would be safe, but then I have to go back and do all the preclinical safety tox study in order to convince regulatory authority.
Okay, great. Okay, so it's a practical issue about what we know about human studies and safe doses of carnitine. Okay,
Exactly.
Got it. And then the glucose you have to add in because of what?
Well, first of all, to compensate for the, let's say, reduction of xylitol, to keep xylitol at a relatively low concentration, let's put it in this way. So, thus, glucose still has some benefit. We should not forget that there's a nutritional component.
Okay. And then my next question, is there a caloric load to xylitol above and beyond just the glucose component? What do we expect the caloric load to be for patients?
It would be much lower than glucose, and you match for the concentration. And then it will mainly follow the pentose monophosphate shunt. So from the caloric standpoint, it's a very poor, let's say, caloric provider.
Okay. So it's not a significant source. So most of the calories from the solution come from the glucose. Okay. What about the transport characteristics of xylitol and carnitine relative to glucose? Is it absorbed with the same speed if we control for membrane characteristics and membrane function?
Absolutely. There is a key international paper where we follow the PK of carnitine provided through an IP route. And it's interesting because carnitine there, it will be immediately absorbed. It will really suck it up. And after the first 2-3 exchanges with carnitine, then eventually it will reach a steady state, an equilibrium, not with the concentration, not really with the concentration that you have in the PD cavity, but at an intermediate level. But if you continue the treatment with, let's say, carnitine, you will stay up to, let's say, with the 0.02% that we are using, we will reach a plasma exposure of carnitine of 300 micromolar, which is 6 to 7 times higher. And this is what we want. We want to overexpose our insulin target organ to carnitine in order to, let's say, trigger the favorable metabolic effect.
So now that we know a little bit more about the solution, and we'll come back to it, maybe, Richard, we can ask you, as one of the sites that's running the clinical trials, which is very exciting, tell us why you wanted to participate, and maybe if you could tell us a little bit about the trial.
Yeah, thanks, Jeff. I think for all of those who are doing PD day to day, including hopefully many of the listeners, we all recognize the challenge of using glucose-rich solutions in our day-to-day practice. I think the problems with ultrafiltration, problem with long dwells, the issues with GDPs and heat sterilization associated with that, and both the metabolic implications and the peritoneal membrane implications of using high glucose solutions, I think are well known to us. It is really exciting to see new solutions coming into the research area and potentially having a clinical impact. And that's why we have been an enthusiastic site recruiting people into this study. So this study is a CAPD study, and I think it's worth reflecting that this is a phase 3 study looking at basically a regulatory an endpoint, trying to show non-inferiority against a glucose-containing solutions. All patients in this study are using an icodextrin-based long dwell, and then the Xylocore solution is being used for between 1 and 3 of the short daily exchanges during the daytime. So it's a randomized one-to-one randomized control trial phase 3 study with a primary endpoint, which is a KTOV of 1.7, and looking at basically non-inferiority. Now, I think, Jeff, you and I and Nikhil would all recognize, many people on this, listening to this podcast would recognize the trouble with trying to use endpoints where we're using small molecule clearance as our primary endpoints. But I think we have to reflect this as a regulatory study trying to show non-inferiority and safety.
Do we go on the record that say you and Edwina's unit are measuring KT over V?
Yes. Obviously, I work alongside Edwina Brown, Professor Edwina Brown, who will be known to many people listening in. And I don't think either of us would argue that we should not measure KT on V. The point about KT on V is as Jeff, it's only one part of the holistic assessment of the person doing PD at home. In this study, this is a primary endpoint to demonstrate non-inferiority against currently available glucose-based solutions. I think the exciting area is going to be the secondary outcome data and the long-term outcome data that shows the sort of cardiometabolic effects, but also the effects in the peritoneal membrane. So I think, yes, the primary endpoint may not be the key bit for us as clinicians that we're interested in, but actually if we can then get this into either secondary outcome measures to be interesting or longer-term studies demonstrating some of the interesting effects that Arduino has hypothesized will be where I think the scientific and clinical benefit will lie.
So when you say, so everybody's using a long dwell of icodextrin, and you set up from 1 to 3 exchanges of Xylocore. A couple questions about that. Is it, if they're on 3 additional glucose exchanges, they would all be Xylocore? Is that because not all of your patients are doing 4 exchanges? Is that why you said 1 to 3? But nobody's going to get glucose solutions in the Xylocore arm, correct?
Absolutely. So those individuals randomized to the interventional arm, their short dwell changes with glucose, the number of exchanges there are replaced exactly for either a low, medium, or high osmotic strength Xylocore solution. So the interventions in the interventional arm receive none of the conventional fluids apart from the icodextrin. Those in the control arm receive their standard glucose exchanges depending on what they were on before. The study requires investigators to modify the dialysis prescription to ensure that KTMV is kept above 1.7. There are people who've gone in the study from two short daily exchanges plus icodextrin to 3 daily exchanges plus icodextrin to ensure that the primary outcome measure will be met.
Got it. Okay. Couple more questions, because this is really interesting. This may be dovetailing into Nikhil, and I think Nikhil might also be getting at this. Can you manipulate the tonicity of the solution? So with glucose, we have 136/15, 227/25, 386/425 hydrous versus anhydrous. Does Xylocore come in different concentrations to manipulate? And what's being manipulated? The extra glucose to do that? Or all 3 are being increased in tonicity?
Arduino, do you want to respond to that, or should I comment?
If you wish, I can respond to this. The first 2, the most common PD solution used in Europe, at least, the low, the equivalent of 1.5, and the intermediate, the 2.5, there, the only, let's say, change in concentration occurring of the 3 is on the xylitol. The xylitol will go from 0.7% up to 1.5% for the intermediate, keeping both glucose and carnitine at the same concentration. Carnitine 0.02% and glucose 0.5%. Then, if we need to treat the patient with the Niger osmotic strength, then we increase a little bit more xylitol up to 2%, and then we increase glucose to 1.5%. That's the idea. So only for the higher strength, we will modify both xylitol and glucose.
That's excellent. So I'll ask a question next about the ultrafiltration part of it. Yeah, go ahead.
I was just going to say, to describe the technical background to that. The clinical and patient-related impact is that there is essentially a low, medium, and high strength, which corresponds to a low, medium, and high strength glucose-containing fluid. And when we're converting people into the study, we're just using directly taking people on low strength back to low strength and medium to medium where they are on those. Sorry, go ahead, Nikhil.
No, that's perfect. I was in the same area. So if I needed a patient who had a requirement for more ultrafiltration, I would use these as glucose is being used currently. My question is simple. Is there any data on how much ultrafiltration is achieved? And is it comparable to what, in a similar kind of a patient, comparable to what glucose achieves in those particular concentrations?
We have done a phase 2 clinical trial in which we were using either the low or the intermediate. So these 2 are the main one. And we have tested these 2 in a 1-month single-arm clinical trial, and we were getting the same ultrafiltration, let's say, efficacy that when they were using glucose. So we didn't see any major difference. The same Bazzato in 1982, and there he was using even a 3% PD solution, only xylitol 3%, which was replacing, let's say, the 2.5%, the 3.86, whatever, 4.2%, the highest osmotic strength, and he was getting a similar, let's say, ultrafiltration efficacy.
There is clearly secondary outcome measures, which includes 24-hour peritoneal filtration and diuresis. Arduino's built up a reassuring evidence base around this to come into the phase 3 study. The phase 3 study is not only going to answer the question about KTMV, but it's clearly got built within it key secondary outcome measures, which I think is where your concern and my concern would be about loss of residual kidney function. Are there more rapid changes in residual kidney function, 24-hour peritoneal ultrafiltration and diuresis, as well as some of the broader metabolic questions that where the interest of these fluid lies.
Yeah.
That's really this. And then, so right now, how many patients have been recruited and over how many?
We should be recruiting 170 patients according to all the statistics, evaluation, calculation, etc. So far, we have 68 patients randomized.
Wow. That's
right. Not that bad. 22 patients completed the study so far. And I have some good news that I can share with you. One patient, after 1 month of treatment, because the short-term glycemic control was that good, after 1 month of treatment of Xylocore, they removed completely insulin. And he stayed without insulin for 5 months. Then when he started again using, because at the end of the study, he was back to the physiolil, all right, he was doing 2 exchanges with 1.36%, etc. The second day of a glucose-based treatment, the short-term glycemia was not under control anymore. They implemented insulin therapy. The other 2 diabetic patients, after 1 month, they reduced the dosage almost 3 times.
Are you hypothesizing that's because of not using glucose, or effect of using less glucose plus some of the beneficial effects?
You need an anti-diabetic action there. Not really a glucose-sparing effect, but an insulin-sparing effect. And then you have to admit that there is an anti-diabetic action, which is carried out by both carnitine and xylitol. It's a concerted action. Plus, xylitol is also limiting insulin secretion, which is good, if you wish.
That's really interesting. Let me ask you guys a couple of other rapid-fire questions. You're measuring hemoglobin A1C in the study. How long is the study? How long are patients?
6 months.
6 months. Are you doing PET pre and post?
Yes, there is a PET. PET is part of the secondary outcome.
Okay. And CA125, just for what it's worth? Is that being measured?
I don't think so. We have done CA125 on the previous study.
All the low GDP neutral pH studies were like a CA125 explosion of studies.
Then we have published several in vitro papers showing that our solution compared to the traditional one is much more biocompatible, whatever. But we are preparing now a big paper, because we have also looked at, and you probably may have heard, the Francesco Trepiccione team. They have developed this multiphoton stuff to look at fibrosis in vivo in rodent exposed to either glucose-based solution or Xylocore and so on and so forth. We can measure the vascular thickening of the peritoneal membrane and so on and so forth.
Okay. I think, Jeff, what I would say is this isn't, as a clinician, the clinician's take on this, the simplest the clinician's take on this isn't about glucose minimization. It's about potentially glucose minimization, insulin burden reduction. And it's not just about the peritoneal membrane. It's about the broader cardiometabolic effects associated with the solutions we're currently using.
Exactly.
And I think there's some really exciting data, preclinical data about fibrosis in the peritoneal membrane and the effects of xylitol. I think these are all positive ticks. It's not just about avoiding glucose. There's some other positive ticks that might be manifest.
Couple quick questions to both of you. There's rapid fire questions. So we talked about CA125. Are you measuring dialysate IL-6? No.
We will collecting blood or plasma sample, urine sample, dialysate sample, centralize all the sample. And in Denver, there is a great guy, Angelo D'Alessandro, is leading a metabolomic, proteomic stuff. He will be analyzing everything.
Okay, so that's great to know.
Fine. So we will have all the data.
Of course, we're collecting information on peritonitis events.
Yeah.
And my one other question is the buffer in Xylocore right now is a lactate buffer.
Lactate base.
Is there any reason it could not be a bicarbonate-based buffer?
Is there any reason why we should not use lactate?
No, that's a whole new podcast. That's another podcast.
But bicarbonate, when you fully metabolize bicarbonate, you form water, and you are using significant amount of bicarbonate. And from bicarbonate, you form water straight away. And if you may remember, many years ago, one of the first studies in which they were testing this carbonate-based PD solution, there was some issue with, if I remember correct, on water retention and so on and so forth.
Yeah.
If you make some calculation, you will be forming a lot of water with
I
bicarbonate.
guess just the infusion pain and the pH. What's the pH of Xylocore?
The pH of the current solution is neutral pH. But we will go back when we sterilize this solution with 0.5% glucose, with on average 5 times less than glucose. The GDP production, even in an acidic environment, slightly acidic environment, which should promote glucose autooxidation, will be very low. Plus, you have xylitol and carnitine, which will moderate the autooxidation of the formation of glucose degradation product. We have tested some of the, let's say, analog of this glucose degradation product, like 2-deoxyglucosone and so on and so forth. And the amount present there is comparable to the physionyl with the lactate bicarbonate mixture and so on and so forth.
Okay.
So we are thinking, because having a single compartment, it would make our life much easier.
Okay.
That's it.
2 more questions for me, either for you or Richard. Is there any reason, theoretically, we couldn't use this as an APD solution? Of course, on our side of the pond, we're a lot heavier users of APD. There's no reason we couldn't be using the solution for APD, correct?
I don't see a reason why not. No, not at all, Jeff. This is it's easier to deal with as regulatory in the CAPD study, but there's no reason why this couldn't be used as a short-term APD solution.
I would have liked to run this study even on APD patient, because I could have, let's say, maximized recruitment, because CAPD in Italy or in Europe, you have a 25 to 30% of the patient in CAPD. All the rest are in APD. But we have also another solution in development, which is the B model. We have polydextrin, 4% of polydextrin plus xylitol and carnitine. This solution is it's one of my favorite one, all right? It's really my favorite, because this will be, Jeff, right now with this clinical trial, we are paying a big, let's say, penalty, because we are losing a lot of carnitine and the xylitol through the icodextrin exchange, because the icodextrin exchange will remove those metabolites or those ingredients that should exert their favorable metabolic effect. That's why I want to have my polycore, in which you keep constant over time the concentration of xylitol and carnitine. Otherwise, if you have this interval with icodextrin, you will be cleaning the body of.
I want to say also to Richard, one of the things that I think about now is that I think research in PD, as we think about modern PD, is very much about patient setting priorities and patient engagement. So I just want to put a plug out that I think this is exceptionally exciting. I think future research, and I want your perspective on this, Richard, should definitely have patient engagement and what's important to them in their PD therapy. And we know that there's been fantastic initiatives like the Standardizing Outcomes in Nephrology, that have established these really important core outcomes to the PD community. So I'd say so often we have a sort of vague evidence base to support a lot of what we do. Like a good example in nephrology is phosphate binders and phosphate, which the evidence base is poor. I really hope that we take this data, if the study is favorable, which we're all crossing our fingers that it is, and if we commit it to clinical use, that we don't just implement it and hope for the best, that we really think about patient engagement in terms of the types of studies we design in the future, and committing it to Xylocore to some of the more harder clinical endpoints that really matter in PD. What are your thoughts about that, Richard?
Jeff, I totally agree. I think you and I both recognize the benefit of co-designing studies with people who've got lived experience around this. I have to say, to get this, I've not been involved in the setup and delivery of the study, to Arduino to get this to a phase 3 study has been a phenomenal amount of work. It's not just the manufacturing that Arduino has hinted at, but the logistics of supply within country, the flux of the investigation of medicinal products has taken many years to sort out. And I think some of those challenges are easily overlooked. Nonetheless, it would be really good to have really strong patient engagement at any stages in these studies. And I think you're right, as the next round of studies around these fluids really should have strong patient engagement if possible. Nonetheless, it's very clear to me that if I sit in clinic with people who I look after and say, look, are you interested in a study where we might be able to reduce your exposure to glucose? Most patients get that, want that. They recognize the harmful effects associated with glucose and sign up to that. That's not to say that we shouldn't be co-producing studies and clinical research, but it's not as though this is not aligned with what I think most people want to be doing.
I'm really happy to hear that. I'm really happy to hear that.
One last question from me. I trained in India. Cost is a big issue, obviously. Local manufacturing of PD fluids is something that is being considered. What are the cost implications if this kind of goes forward in such a product?
Xylitol is not cheap, but now it's becoming more and more popular, particularly in the nutritional field. The real issue is to find somebody able to provide you with the API, because for this kind of product should be an API, possibly pyrogen-free, and so on and so forth.
I lost my train of thought because you're so passionate, Arduino. It's hard to think when you're talking because you're just enamored with your passion. I was gonna ask about you're measuring oxalate as well in the study.
Yeah.
Oh, because that makes sense to be measuring.
I think Arduino came up with the comments about xylitol to oxalate transformation. I went to go and while we've been having this conversation, remind myself what percentage is converted. It's less than 0.02% in clinical studies. So I think the safety concerns, of course, it's right to think about the safety concerns and some of the aspects that, but it's a pretty minimal conversion rate.
And then my final question, is there any patient or study subject that you would be a contraindication to use of Xylocore solution?
Please, Richard.
So the key exclusion at this stage is people carrying primary hyperoxaluria type 1 genetic polymorphism. I'm allowed to share this. The irony of this is that PH1 primary hyperoxaluria type 1, as we recognize, is a rare genetic mutation associated with usually kidney failure in young people and often renal, cardiac, and central nervous system problems. The first slightly older patient we screened into the study transpired to be a compound heterozygote for PH1, and we therefore had to exclude from the study. So the first patient in the intervention arm found out, but it is people carrying PH1 mutations who are at the moment excluded from this other key exclusion criteria from the study.
Are you screening for that, or is it just?
Yeah, so if you're the study will comment more.
Oh, you go ahead, Richard.
In the individuals randomized to receive the interventional arm of the study, they undergo PH1 genetic screening before receiving the xylitol compounds, and that clearly needs to be thought through and resolved if this proves to be as safe and as effective as one might hope it would prove to be, and think about then how we might operationalize that and the safety mechanisms in wider clinical practice and further studies.
All right. I think we are at time. We are beyond time, but that's okay. This was a passionate podcast. I'm loving it. And I'm just going to say, I might even title it Mamma Mia. Who knows? Thank you so much for this absolutely exciting product that is being tested. And I honestly wish that we could help. We could participate too someday in something like this.
I want to visit Jeff and discuss, because now that I'm getting excited with this type 2 diabetic patient, why don't we do a phase 2, phase 3 a superiority study to show that we can improve glycemic control by reducing insulin treatment, so on and so forth, in Canada?
Yeah, no, I think there's definitely far-reaching benefits to think about. And I was going to say that when we have Jewish New Year, we usually wish people a sweet New Year, which is to have a sweet and healthy New Year. And now that we start 2025, this podcast is not wishing us as much of a sweet New Year. We want a glucose-sparing New Year, perhaps. With that, I just want to thank you and Arduino and Richard on the clinical side for really getting us excited about PD. And it's nice to see innovation and advances in PD and the field moving forward. And I think, if your science is met with your passion, then I'm sure it's a great combination for ongoing success. And I want to encourage all of our listeners to look at the PDI issue about the ELIXIR trial. And we look forward to seeing ongoing developments as the trial progresses. Any final thoughts from you, Richard?
at Arduino?
Jeff, I'd just echo Arduino's enthusiasm, but as you said, it's really great to see new fluids coming into research studies, into phase 3 studies. We haven't seen anything really since in the last 10, 15 years that have innovated and provided novel fluids in phase 3 studies. So I think Arduino's has a great success getting this far, and hopefully we'll see some benefit from this study.
Yeah.
One final thought. I learned so much with the PD. Guys, I'm 71. I'm not really a young kid. And I have recovered a lot of what you say, passion and strength. I want to go back to an insulin-independent glycemic effect, glycemic control, insulin-independent. Since the very beginning, I was thinking, and PD is giving me the opportunity really to test this. I'm testing this hypothesis, if you wish, in human.
Wow.
Really well.
That is absolutely fascinating.